Hi everyone, a very good evening to one and all of you. Today I will be taking yet another topic from recent advances in hematology, third edition, and that is a very small topic that is thrombin activable fibrinolysis inhibitor. Okay, so today we will be discussing this topic under the following headings, that is the role of TAFI in regulating fibrinolysis, its potential as a therapeutic target for thrombotic disorders. See, as the name suggests, it is a fibrinolysis inhibitor. So, what is fibrinolysis does in our body? It is a pathway that lyses the clot or the thrombus, which is formed by the platelet plug. So, when the clot is lysed, then the thrombus is gone. Okay, so if the clot lysis is inhibited, there means there will be so much of clot formation which is not lysed. That is, there is thrombotic disorder. Okay, so when TAFI is increased, that is clot lysis is inhibited, clot is going on forming. So it is a thrombotic entity in the patient, and then potential as a therapeutic target means we are targeting TAFI in thrombotic disorders to give a treatment to the patient. And then we will discuss about the TAFI, its properties, its epidemiological studies, its genetic variations, and the potential inhibitors of TAFI that can be used as cure for thrombotic disorders, as well as the significance of TAFI in diseases. So, as you all know, TAFI is thrombin activable fibrinolysis inhibitor. So, it is by function inhibits fibrinolysis, as I said, and is activated by thrombin. Okay. It is also known as procarboxypeptidase. Why it is known as procarboxypeptidase is that TAFI was first reported by Hendricks et al., but in its structure, it was purified and characterized by Yaten et al. So, Yaten et al. Noted that the pancreatic carboxypeptidase enzyme, it has the same chemical structure as TAFI. So, that is why TAFI was also known as procarboxypeptidase B, and it is a plasma zymogen. So zymogen is an enzyme which is in its precursor version. That is, it should be activated by something else, some other protein, then only it will be converted into its active form. Okay, so here it is activated by thrombin. So another zymogen which we know is trypsinogen from the pancreas. You know, the pancreatic enzymes, they should never be in the active state in our body, then all our organs and everything will get lysed so fast. Okay, so they are very strong lytic enzymes. So they have to be first activated by trypsinogen, chymotrypsinogen, etc. So it is activated and converted into its active form inside the small intestine, and that is how it acts in the pancreas. The pancreatic enzymes are acted in the intestine. So here TAFI is a zymogen which is activated by thrombin. Okay, and then Badzer et al. Identified its antifibrinolytic effect. So, and then he gave this designation that it is a fibrinolysis inhibitor, which is thrombin activated. Okay, so how does it cause fibrinolysis inhibition? Is that it removes carboxy terminal lysyl and arginine residues from the partially degraded fibrin. Okay, the degraded fibrin, which is partially degraded fibrin. So every protein has a carboxy terminal and amino terminal. So in the carboxy terminal, the lysyl and arginine residues or the amino acid are removed by the taffy, and then that is how it causes inhibition of fibrinolysis. But its main function is that it inhibits plasminogen activation. By doing this, so it inhibits the plasminogen activation. How does it inhibit plasminogen activation by removing the terminals from the fibrin. Okay, so from the fibrin, when these residues are there, then only plasminogen can get activated to cause fibrinolysis. Okay, that is an innate mechanism in our body to for because the clot should not be anywhere permanent. So to remove that, it is an innate mechanism that it will activate plasminogen and it will cause fibrinolysis. But TAFE inhibits this by this function. So what is fibrinolytic pathway is an endothelial cell when there is clot formation, then the thrombin activates protein C also. So TAFE is also activated in the same way protein C is also activated by thrombin, and this activated protein C with the help of protein S, and it inactivates 8 and active 8 and fifth clotting factor, and by this way it inhibits clotting pathway. Along the same way, it also activates plasminogen to plasmin, and the plasmin lyses the fibrin. Okay, this is how fibrinolytic pathway happens. So here what happens is that, as I said, thrombin activates protein C. In the same way, thrombin also activates TAFE, and then this TAFE will now inhibit the conversion of plasminogen to plasmin by removing the allyl and arginine residues in the carboxy terminal of the fibrin in the clot. Okay, so that is the function. So what are the properties of TAFE? TAFE, it is synthesized as an amino acid pre-pro-peptide. I said it is a zymogen. Before that also, before becoming a complete peptide only, it is formed as pre-pro-peptide with 424 amino acid. And then this pre-pro peptide includes signal peptide, activation peptide, as well as the catalytic domain. Okay. And then the mature protein has 401 amino acids only, which has a molecular weight of 45,999 Dalton. And by glycosylation process, the protein weight increases to 60,000 Dalton. Now, after that, again, when it gets active, this is a pro-protein only, okay? When that is that plasma zymogen form. In the active TAFE form, the molecular weight is only 35,000 Dalton. And then Buffer et al. Had synthesized a recombinant TAFE which exhibits the same enzymatic and functional properties of plasma-derived counterpart of the TAFE. So it was laboratory created recombinant version for research purposes mainly, and they could also identify with the help of this recombinant TAFE the location where the thrombin cleaves the TAFE and it makes TAFE to the activate form. So now coming to the epidemiological studies and TAFE. Plasma TAFE concentration has been studied by various scientists to check what is the plasma TAFE concentration in different individuals. Okay, and they reported difference of up to tenfold. Okay, the studies have shown varying levels of TAFE antigen that is ranging from 4.4 microgram to 15 microgram per ml. Okay, and it is attributed to the assay which they have been used to identify the TAFE, and then different kit reactivities, that means the kit which they have used to quantify the TAFE. And then there are TAFE isoforms as well. Okay, so you know ki creatinine kinase in our body has isoforms like MB, BB, etc., as which is seen MM in the myocardium, BB in the brain, etc. So in the same way enzymes have isoforms, in the same way TAFE also has isoforms. So in some individuals, isoforms are certain type of isoform which the scientist has used for study. Maybe they have taken one type of isoform and all the studies were done, and those may be having a range of 5 to 6 microgram per ml for every individual. And the other study they used different isoform and they found more increased TAFI antigen level. Okay, so it depends upon the isoforms in which the scientists have studied also. A low or high um TAFI plasma concentration may influence the balance between pro-fibrinolytic and anti-fibrinolytic pathway. Obviously, it is a fibrinolysis inhibitor. So when it is increased, so it is anti-fibrinolytic state of the body, and if it is decreased, pro-fibrinolytic state of the body. And according to that, it can be a bleeding state or a thrombosis state, that is hemorrhagic state or a thrombotic tendency to the patient. So studies have used to correlate that with the stroke patients, the DVT patients, and coronary artery disease patients. But sometimes the studies has indicated contradictory also. That is, if you are thinking in deep vein thrombosis, it is a thrombotic state. Okay, so obviously TAFI concentration should increase because TAFI is inhibiting fibrinolysis, that is clot is not getting lysed, so there is increased clot formation tendency or thrombotic tendency. So in DVT, obviously TAFI should increase, but certain studies have shown decreased DVT also. So that is not correlating, so that is contradictory. So certain studies are in favor, certain are not also. But many studies which have been done in stroke patients by Montaner et al., Santamarie et al., Liebeck et al., and Biswas et al., etc., have showed increased TAFI antigen in ischemic stroke patients. Ischemic means blood circulation has gone stopped to that part of the stroke. Ischemic stroke, that means in brain certain part the blood circulation has been stopped, and that is also especially caused by thromboembolism. Okay, so in certain those conditions, increased TAFE is actually favoring the hypothesis that increased TAFE is associated with stroke. Okay, and the study was done within 24 hours of the stroke patient. The serum was collected and TAFE was done. And within 1 month of the event, also when it was done by Santamarie et al. In young ischemic stroke patients as well as in transient ischemic attack patients, in that also it showed increased TAFE level. Okay, and other studies also has contributed to the same. So increased TAFE level may be associated with stroke patients. So if we so now coming to single nucleotide polymorphisms in the TAFE gene. The TAFE gene is located on the 13th chromosome, that also in the short term. And when first the coding polymorphisms were described, the polymorphism that was found was G to A transition, that is guanine to adenine transition in the 505th position. Okay, so in the gene there are codons, which I said there are 424 amino acids. There may be 424 codons that code for the amino acids. Okay, so each codon has three nucleotide pair as we have studied before. So taking 424 into 3, that many nucleotides are present in the gene. So in that 505th position में when guanine is replaced by alanine, okay, guanine is replaced by adenosine, sorry, the amino acid that is alanine get replaced to threonine in the 147th position. And other polymorphism that was identified was cytosine is changing to thymine in the 678th position. But this is a silent polymorphism because the codon which is changed to another codon, and this both codon are coding for the same amino acid. So it is a silent mutation or a silent polymorphism, you can tell. So these were the first described polymorphisms. Other than that, seven new polymorphisms have also been identified in the promoter region or the untranslated region of the gene as well, which has caused increased TAFE gene level or how called its thermal instability and stability, etc. Then key contributors, when we have done multivariate analysis, not we, the scientists, when they did multivariate analysis, the key contributors that is the most common polymorphism that was identified was in 1542nd position, cytosine was replaced by guanine, and in 147th position, as I had said, of the amino acid, alanine was replaced with threonine. That is 505G to A transition. That is a polymorphism that has been found. Most common, both. Then coming to how the regulation of TAFI gene expression is, I had said key in promoter in and in untranslated regions there are polymorphisms that affect the TAFI levels as well in every individual. When there is this polymorphism, the levels are different. And in population studies, these findings were consistent with the Brazilian and the African populations as well. That is, some untranslated regions where the polymorphism, certain type of polymorphism is seen, then those people have increased TAFI level, and other type of polymorphism, they might be having lesser TAFI levels, etc. So most polymorphism studies have focused on patients with MI on R or arterial thrombosis, and the recent studies, including the stroke population also, they confer that 3 untranslated region polymorphisms regulate the TAFI antigen level in an individual. And in Asian Indian population also, the TAFI antigen level has been seen associated with the single nucleotide pleomorphism. Now we can see the inhibitors of TAFI. Okay, if increased TAFI level is leading to stroke, then can we use the inhibitors of TAFI as a treatment for stroke patients, or as prophylaxis, or to prevent further stroke attacks to the patient? So, since TAFI's catalytic action requires zinc, so when zinc is used as a chelating agent, it can inhibit TAFI. So, these chelating agents that chelate zinc, like O-phenolphthalein and EDTA, can be used as an inhibitor of TAFI. In the same way, reducing agents like thiotol and 2-mercaptoethanol that reduce disulfide bonds also can be used, and natural inhibitors like PTCI, potato tuber carboxy peptidase inhibitor, can also be used. Okay, so summarizing the whole thing now, so I said TAFI gene is located in the 13th chromosome in the short arm, and it shows many single nucleotide pleomorphisms which affect the TAFI serum levels in the individuals. And then PTCI, as I said, it is a potato derived TAFI inhibitor, which has 39 amino acid, which can be used as a TAFI inhibitor. And what is TAFI? It is thrombin activable fibrinolysis inhibitor, or also it is called carboxy peptidase B2. And it inhibits fibrinolysis by cleaving the carboxy terminal lysine residues on the partially degraded fibrin. Now, it plays a very important role in prothrombotic disorders like stroke and then coronary artery disease, and potential inhibitors of these can be used as attractive therapeutic targets. So, take home message is that TAFI is involved in regulating fibrinolysis, and it is an attractive potential drug target for treating thrombotic disorders. Future research should be focused on developing newer inhibitors for these thrombotic disorders. So we come to the end of the presentation. I extend my gratitude to Mr. Arijit Biswas, Mr. Suhail Akhtar, and Ms. Renu Saxena for contributing this beautiful article in Recent Advances of Hematology, third edition. Kindly do like, share, and subscribe this YouTube channel and comment your questions and the topics which you want me to take. Thank you. Bye.